Here's the answers for the final video. For 1A, you got the N-terminus, and then bracketed to the C-terminus, you got the NH3N, H3N plus, bonded to the C, bonded to the H, going through that whole deal. For 1B, primary uses peptide bonds, secondary hydrogen bonds. I'll be putting in class that's secondary hydrogen bonds, tertiary hydrogen bonds, ionic bonds, diffuse bridges, and hydrophobic interactions, quaternary hydrogen bonding, and ionic bonding. Primary structures are held together by covalent peptide bonds formed between amino acid in the polypeptide chain. Secondary structures use hydrogen bonds between the amino and the carbon group of the polypeptide chain, allowing it to form alpha helices and beta sheets. The huge amount of interactions within the territory oh my gosh, I can't speak. Structure allows it to be a 3D shape. These shapes determine a protein function of determines how it acts with other molecules. The non-covalent interactions used in the quaternary structure allows multiple subunits to come together to form a larger protein complex. For C, two types of enzyme subtract interactions, including the lock and key, and then including the fit. In the lock and key model, the enzyme acts as a lock and has an active site which only a particular subtract can bind, which could be our key. Subtract, enzyme, subtract. The subtract fits in there perfectly. The second is an induced fit. In this type, the enzyme and subtract undergo structural change to the atom and a optimal fit. So you got the subtracts, active sites, enzyme. The subtracts go in the enzyme complete, and just walks them through the stages. For 2A, you got the phosphate group, the pentose sugar, and the nitrogenous base. For 2B, DNA double-stranded contains sugar. Deoxyribosome uses thymine T. RNA single-stranded contains ribosome instead of deoxyribosome and uses the nitrogenous base uracil or U rather than thymine. DNA is used to store genetic information, so it's as a long-term storage of genetic codes, while RNA acts as a messenger and is only a temporary code while the specific portion of a DNA, which is used for protein synthesis. DNA is also double-stranded, making it move stable and longer-lasting, while RNA is a single-stranded and easily broken down after it has been used. 2C. A virus is made up of a DNA and RNA genomes that is found within the virus capsid of protein shell, and sometimes it also has a lipid envelope. There are envelope proteins, also referred to as aspels, that allows the virus to attach the receptors on the host cell membrane and then inject the genome into the host cell. When a virus attaches its connector, the host cell via fusion, endocytose, or direct injection, one inside the capsule is broken down, allowing the genome to be transcribed and translated by the host cell machinery. Once this is done, the newly made virus makes new virions, which are released by the host cell via budding or cell lysis. 3C. In shorter terms, anabolis has to do with the building up, Catabolus is used to break down. Anabolus uses energy from ATP or NADPH to create larger, more complex molecules from smaller ones. Anabolus includes protein synthesis to chemical reactions that use anabolus are condensation reactions and dehydration synthesis. Catabolus uses the breakdown of a larger molecule into smaller to release energy to major types of chemical reactions, including hydrolysis. 3B. ATP plus H2O, the higher, yields ADP plus PI plus energy, which is lower. This is an exergonic reaction because energy is released and the delta G value would be negative. 3C. When you eat starchy foods, the mouth produces saliva containing the enzyme salivary amylase, which is used to break down the starch into dextrins and maltose. But as the food continues down the digestive tract, the small intestine uses pancreatic amylase to break down the fragments into even smaller discahydrides like maltose. Then the final step of breaking down occurs in the intestine lining, where maltose splits maltose molecules into individual glucose molecules that are absorbed into the bloodstream through the interstitial wall. Thank you.